Good morning. Thank you for inviting me to speak at this workshop. Indeed, I am an independent consultant, and I'm speaking currently as a member and organizer of the CTAC Pelston workshop, which was actually held last year in February, and all the papers appeared early this year. So the next slide, please. We came together with 48 global experts, equal representation from government, academia, industry, consultancies, and we actually did achieve consensus, which we were very happy to do, and we addressed the controversy at the heart of environmental safety assessment of endocrine disrupting substances, and I'm going to tell you how we did that. Obviously, the views and statements are not those of the organizations necessarily, but of the participants to the workshop. And we explicitly avoided human risk, realizing fully that in ecotoxicity testing and evaluation, we're looking to protect populations by looking at the impact on individual wildlife. And of course, human protection goals are different and much more stringent. So we did invite mammalian toxicologists to inform our discussion on environmental evaluation of endocrine disruptors, but we did not touch on human health. Just to make that very clear. Next slide, please. We had, as I said, reasonably balanced representation from all the different sectors, and also quite a broad global representation: North America, Europe, Japan, Australia. And next slide, please. Just briefly, for those who are not familiar with CTAC, CTAC is a not-for-profit global professional organization bringing together 6,000 members from around the world, with a balance from academia, business, and government. And the basis of the organization is science-based objectivity as a key founding principle. And then the next slide, please. So, as you are all aware, the continuing scientific concern, public debate, media attention over endocrine disrupting substances that are or have been present in the environment at concentrations harmful to wildlife, jurisdictions initiating regulatory approaches, a lack of consensus, some scientists believing that EDSs can reliably be assessed using standard risk assessment paradigms, and others that do not believe that this is sufficiently precautionary and propose risk management on the basis of hazard alone. Next slide, please. Led us to try to find scientific agreement, which, as I said, we achieved, on how best to assess the impact of endocrine active substances on wildlife. We hoped to, and did indeed, provide guidance, assisting regulators, registrants, policymakers in their decision-making, contributing to speeding up the global progress in controlling these chemicals, and identifying areas of, yes, uncertainty and knowledge gaps. So in the next slide, I will tell you a little bit about, next slide please, how we organized the workshop. We worked on 6 case studies. So the 48 members were divided into 6 different groups. And leading up to the workshop itself was held in February 2016 in Pensacola, Florida. But leading up to it, starting from September the year before, each of the groups worked on a case study. And we selected endocrine active materials. The materials we worked on were 17-alpha ethinyl estradiol, an estrogen agonist, perchlorate, a thyroid agonist, propiconazole, a steroidogenesis inhibitor, tributyltin, retinoid receptor inhibitor, vinclozolin, an androgen antagonist, and 17-beta-trembolone, an androgen agonist. So we tried to really cover a very broad range of modes of action of endocrine disrupting substances, endocrine active substances. These were all considered to be data-rich in relevant information for one taxa in the case of EE2, which I'll explain in a moment, or more ecologically relevant taxa at multiple levels of biological organization, from the biochemical level to the whole organism level, and sometimes to the population level. And a comment on data-rich and several taxa. As I said, EE2 focused on one taxa, focused on fish. The reason being, first of all, the aquatic environment. We had to apply some kind of focus, because in the case of EE2, we're looking at 15,000 papers, which we brought down to 1,371 to be evaluated or to be assessed for their relevance. And the focus was on fish. We had too many data available, and it was clear that a vertebrate taxon would be most suitable here with estrogen receptors. And so that's for EE2. And for perchlorate, we went from 7,000 references to about 180. A focus on, first of all, reproductive developmental studies, because development is a sensitive life stage for thyroid hormone insufficiency. We focused at population level effects to see how survival and reproduction, growth and development effects impacted at the population level, and studies that examined perchlorate mode of action to confirm similar mode of action across species. So that's where several taxa were taken on board. Trembolone, we looked at fish. I'm sorry, for TBT, we looked at fish, mollusks, and other taxonomic groups, including mammals. Here we went from 1,000 regulatory reports and open and grey literature to 160 that we looked at in detail. For trembolone, while we considered in vitro, invertebrate, fish, amphibian, and avian mammalian studies, we focused on aquatic organisms in the end, because for trembolone, that the major route of exposure is via animal waste runoff into aquatic systems. So that's just to help you give some background as to how we applied focus in our quite significant task. Now, another important observation here is that we're not talking about full safety evaluations, but we really went through these case studies to identify as a mechanism for identifying issues that need to be resolved. Next slide, please. So the workflow for the case studies is presented in this slide, and at the top you can see the input of information from regulatory data sets, open literature, fate and exposure data, and we then did a relevance assessment and for adverse and mechanistic information. Non-relevant studies were documented but not worked on further. And then we went to a reliability assessment, which I'll say something about in a moment. Unreliable studies were documented, but the reliable studies were continued with. And then these studies were assembled in a framework to collate data relevant for assessing endocrine axes and really ordering them by different levels of assessment using the OECD framework as a guide in most cases. In some cases that was a little difficult, but in most cases that was possible to use that framework. And then weight of evidence assessment was done, and the approach selected by each group was slightly different. For propiconazole and perchlorate, those groups used a system similar to what US EPA uses. Responses, positive, negative, or no change of each relevant endpoint were tabulated and organized according to interaction with the endocrine axes. And the propiconazole group used the hypothesis testing recommended by Borgert, 2014, to test the hypothesis that propiconazole has potential to interact as either an agonist or antagonist with components of the estrogen pathway, for example, and then each of the pathways. So endpoints relevant for these interactions were considered, and additional endpoints from higher level studies were used as far as they were informative. For TBT and EE2, the AOP adverse outcome pathway based on Ankley et al. Was used to structure the weight of evidence process. So, as you all know, the AOP approach is designed to depict causal link between specific and specific endocrine activity or molecular initiating events and adverse apical endpoints. So back to the slide we're on. Subsequently, this weight of evidence assessment led to a characterization, first of the hazard, and then taking the exposure on board and considering whether a risk assessment could be done. And so that's the approach that we took in this, in preparing for the actual workshop. And then the next slide, please. So just a little bit about the quality evaluation of relevant data. Selected papers were taken through the ToxR tool, Toxicological Data Reliability Assessment tool, which is a common approach for REACH as well. I'm sure most of you will be familiar with it, and it takes on board the Klimisch criteria. So we broadly took Klimisch 1 and 2 to be the guideline for whether publications were acceptable. And Klimisch score 3 and 4 studies were recorded but not used in the further evaluation. And histopath data were addressed more specifically in a parallel exercise, because we found that histopath data, really the evaluation of histopath papers is not given any attention under the Klimisch, or not sufficient attention under the Klimisch approach. So we had a parallel exercise, and the results of that exercise were incorporated in the case study evaluations. So next slide, please. So what I haven't explained yet, after the case studies were gone through, or while the case studies were being gone through, each of the groups were asked to identify cross-cutting issues. So which issues did they run into that led to uncertainty in biological responses and uncertainty in the ability to do a risk assessment? For example, and then we found that we focused on cross-cutting issues that came back in several of the case studies, so that were generally considered as issues by the groups. For example, effects that manifest themselves at a later life stage, following exposure during the sensitive windows of the life cycle, e.g. sex reversal in fish, multi-generational effects reflected in a design of an extended one generation for fish and mammals, transgenerational effects, non-monotonic dose relationships, thresholds, and I want to dwell on that a little because of the relevance to this, to the topic that we're discussing today. So the next slide, please. Sorry for, this is a slightly busy slide. I'm sorry for that. I should have spread that over several slides, but I'll try and go through it quickly. I just want to highlight to you that there were cases of non-monotonic dose relationships and subsequent threshold concerns in some of the, as you can see, 4 of the 6 case studies. Well, the first one is with EE2, a few examples of NMDRs as part of exposure to EE2. Most common ones related to egg production in female zebrafish and fathead minnow. The low concentrations were seen to stimulate egg production, and higher concentrations reduced egg production. And there were also some NOx for fish repro, or absence of NOx in fish repros, because the concentrations weren't low enough. And sometimes that was a result of analytical constraints. However, it was felt by the case study group that there were sufficient fish for life cycle studies with clear NOx and LOX to do a risk assessment on EE2 overall. So individual cases of NMDRs, but no overall concern about doing a risk assessment with EE2. For perchlorate, there were some examples of NMDRs seen. In one study in stickleback, T3, T4 levels were affected at low concentrations, but not at the higher concentrations across life stages. But no apical endpoints were evaluated in that particular case. So that could be a concern. NMDRs were seen in a mammalian 2-generation reproduction study. Increased hormone levels at lower dose levels with a decrease in T4 levels at the high dose, seen at some time points, maybe the result of variability in hormone measurement, or maybe related to the magnitude of the effect, the time of exposure, and the compensatory capacity of the hypothalamic pituitary thyroid axis to reestablish homeostasis. However, the NMDR was not seen at population-relevant endpoints, and therefore, again, it was considered overall by the case study group that there was no impact on risk assessment in this case study. There are further examples, but I think it's going to take too long to go into them, but I would be very happy to expand on them. So the next slide, please. So overall, as I said, NMDRs in the case studies were not considered to impede environmental risk assessment. And in nearly all the cases, the examples on the previous slide of the cases where that was not straightforward, but in all the other cases, when a study was rated Klimisch 1 or 2, apical endpoints, described experiments with apical endpoints and exposure concentration ranges that allowed thresholds to be resolved for adverse population-relevant effects. So we did not consider that there was a concern as a result of these issues in the case studies that we dealt with. And the next slide. However, if there are situations where there is uncertainty as a result of NMDRs, during the workshop itself, so I highlighted that during the case studies, cross-cutting issues were raised. And during the workshop itself, we actually worked on these different cross-cutting issues. And one of the groups worked on how to deal with an NMDR if it does appear. So first of all, this flowchart looks at mechanistic level NMDRs. So is there an NMDR? Yes. Is there a plausible explanation? Yes. Then consider a refined study. And if that refined study addresses the issue, then you can go on with your weight of evidence. Is there not a plausible explanation? Then you wonder, is it reproducible? If not, then you continue with your weight of evidence evaluation, and if yes, then you consider a design of in vivo experiments to address this. And if you go to the next slide, if you have NMDRs appear at the in vivo level, then a similar approach, yes, there's an NMDR. Is there a plausible explanation? No. Is it reproducible? No. Then go on with your weight of evidence. Is it a plausible explanation? Yes. Can a threshold be determined? No. Then do your weight of evidence evaluation followed by a hazard assessment. And if a threshold can be determined, then go on with the weight of evidence evaluation and a risk assessment. And if no is the answer at the bottom, then continue with a refined study. So this is an approach that we felt would be pragmatic in dealing with NMDRs that you do run into. The next slide, please. These are examples of other cross-cutting issues. We felt that the in vitro high-throughput screening to prioritize chemicals for testing and provide insights as to the most appropriate assays to characterize hazard and risk are key. And of course, a lot of those are very far in their development. Adding endpoints for elucidating connections between mechanistic and adverse outcomes, identifying potentially sensitive taxa for which test methods currently do not exist. These are concerns that sound familiar to you all, I'm sure. Addressing key endocrine pathways of possible concern in addition to those associated with estrogen, androgen, and thyroid signaling. And in that context, I just wanted to mention that a couple of months ago, I was in a workshop in Brussels organized by the European Commission looking at data gaps to determine endocrine disrupting chemicals, possibly leading to a funding proposal in Horizon 2020 of next year. So there's all kinds of work going on that topic. So the next slide, more cross-cutting issues at the about population-relevant endpoints. There are examples of population-level impacts from endocrine active substance exposure, e.g. gastropods in the context of TBT, fish in the context of EE2. There's a greater understanding is needed of relationships between mechanistic endpoints and adverse population level effects. Consider the severity and the prevalence of the response. Use newer endpoints, for example, behavior from current newly designed studies. Develop adverse outcome pathways and population modeling that will increase this understanding. And the recovery process should be considered when evaluating population level of effects. Next slide, please. Now, I think I have to apologize here because the writing is a little small. No, but you will all hopefully be able to receive this presentation. In the
interest of time, if you could move on.
Yeah. So this is a decision-making strategy that we came up with. This is really the main outcome of the workshop here, but I would like to ask you to go through it in detail when you see the presentation, because it's very hard to read here. I'm sorry about that. But it takes you through all the steps that you need to consider in coming up with the final answer. So starting with, is the substance an EDS? Will exposure in wildlife occur? Is exposure measurable or prediction reliable? Have sensitive taxa, life stages, endpoints been tested? Are tests adequate to detect the delayed effects? Are tests likely to have missed adverse effects at concentrations below those tested? And then, depending on the answer to those questions, either conduct a standard risk assessment or consider regulating on the basis of hazard until suitable test methods or reliable exposure measures or predictions are available. Next slide, please. So the main conclusion of the workshop was that provided environmental exposure, effects on relevant taxa and life stages, delayed effects, dose and concentration response relationships are adequately characterized, then conducting environmental risk assessment of endocrine disruptor substances is scientifically sound, which is expected in the majority of cases. If data do not allow proper scientific assessment yet, in brackets, a hazard-based decision is scientifically sound, justified. So, next slide, please. Topics of further research, I mentioned them before. They are listed here. I won't go through them again because I've really highlighted them during the previous slides. In the next slide, please. And these are the 6 papers that came out of this workshop, which are all published in IEAM. They were published in March of this year. And I strongly recommend you to go through them, and the supplementary data have all the case studies in detail. So there's a lot of information that will help you be informed about this approach that we've developed at this workshop. And we hope it will help in the current situation as well. The final slide. So again, the take-home message, it is possible, provided all this information is available, to do an environmental risk assessment of endocrine disrupting substances. And in certain cases, if it's not yet available, then a hazard assessment would be scientifically justified. Final slide. Thank you for your attention. And my co-chair, Mike Roberts, and primary author of the recommended approaches paper, Peter Matheson, who was initially approached to give this talk, but he's on his way to St. Petersburg, so I'm happy to give this talk. And there are actually 3 other participants to the workshop in this meeting, which is great. Henrik Holbeck, Steve Dungy, and Katie Cody. And well, that's great that they're here, and maybe they can also help field the questions that come. Thank you.